Good morning, everyone. The topic of today's discussion is anatomy of stems, anatomy of angiosperm stems, that is, the dicot and monocot stems. Coming to the contents: introduction, transverse section of dicot stem, transverse section of a monocot stem, conclusion, references, and MCQs. Now, what is a stem? The stem is the ascending part of the plant axis. Generally, it is aerial in its growth and bears leaves and flowers. Now, coming to the internal structure of a typical dicot stem, that is the stem of Tridax procumbens or the stem of sunflower. So, TS through a young stem appears roughly circular in outline and shows three distinct regions, namely the epidermis, cortex, and stele. The epidermis is the outermost surface layer composed of a single layer of tangentially elongated cells. The cells are compactly arranged without any intercellular spaces. The outer walls of the epidermal cells are cutinized. Some cells bear multicellular hairs or trichomes. In young stem, stomata may be present. If you all remember, in root epidermis or epiblema, there is no stomata, and some cells bear unicellular root hairs, but here the epidermal cells of stem bear multicellular hairs, which we also call as trichomes. Then coming to the cortex. Next to the epidermis, there is a multi-layered region called cortex. It is differentiated into three zones: the hypodermis, the general cortex, and the endodermis. The hypodermis here is collenchymatous. It is made up of collenchyma cells. Then there is a general cortex which is chlorenchymatous and parenchymatous. Partly it is made up of chlorenchyma and partly parenchyma. Innermost layer of cortex is endodermis, which is also called as the starch sheath. So, hypodermis, as I told you, is the outer zone of the cortex and it lies below the epidermis. It consists of 3 to 4 layers of compactly arranged collenchymatous cells with their walls having angular thickenings. The intercellular spaces are absent. The general cortex. The general cortex lies next to the hypodermis, and this is a parenchymatous zone of thin-walled cells with intercellular spaces. These cells may have few chloroplasts. These cells serve photosynthetic in function besides storing a lot of food materials. Then coming to the endodermis, the innermost layer of cortex. It is also called as starch sheath. It forms the innermost layer and usually it is single layered, and in dicot stems it is wavy in outline and consists of single layer of conspicuous barrel shaped closely fitted parenchymatous cells. These cells contain dense starch grains, so that the endodermis is also called as starch sheath. Then coming to the stele. Stele comprises of the pericycle, vascular bundles, pith, medullary rays, and so it is also called as a eustele. Stele here in dicot stem is eustele or a true stele. So these are the pictures of the stem anatomies. That is, the first two pictures show the ground plan and a portion enlarged of a dicot stem. The next two pictures depict a monocot stem. Ground plan, the circular one is the ground plan, and then the elongated one is a portion enlarged of monocot stem. See, you all can see properly in the ground plan, roughly circular in outline, and the three zones of dicot stem: epidermis, and then there is a cortex, and then there is stele. So epidermis, which is single layered, and it has multicellular epidermal hairs or trichomes. Then you can see the cortex, which is differentiated, a differentiated cortex consisting of hypodermal collenchyma, that is just below the epidermis. There is collenchymatous hypodermis, and then there is chlorenchymatous cortex, middle cortex, and then there is parenchymatous cortex. Then innermost layer is a wavy endodermis. Inner to that are the vascular bundles, which makes up the stele. Now coming to the stele. The tissues which lie within the endodermis constitute the stele. The stele is made up of pericycle, vascular bundles, pith or medulla, and medullary rays or pith rays. Pericycle, it lies below the endodermis and constitutes the outermost region of stele. It is partly parenchymatous and partly sclerenchymatous. The sclerenchyma cells, which occur in the form of a patch above the phloem, constitute the bundle cap, which is half moon shaped or semi-lunar in shape, and these cells of bundle cap constitute the hard bast. The parenchyma cells alternate with the bundle cap and develop in the same line. So you all can understand that the pericycle is partly parenchymatous and partly sclerenchymatous. Above each vascular bundle, the sclerenchymatous tissue is the pericycle. And between the two vascular bundles, the parenchymatous tissue below the endodermis is also the pericycle. So pericycle is partly parenchymatous and partly sclerenchymatous. The sclerenchymatous pericycle above each vascular bundle in a dicot stem is called as hard bast. Bast is phloem. Bast refers to phloem. Since the sclerenchyma cells of pericycle are in close contact with the phloem of the vascular bundle, they are also called as hard bast. Then coming to the vascular bundles. These bundles are limited in number and they are arranged in the form of a ring or a broken ring in the intrastellar ground tissue. The vascular bundles here in dicot stems are typically conjoint, collateral, and open. Conjoint because both xylem and phloem are in the same bundle, not like roots wherein xylem forms a different bundle and phloem forms a separate bundle. Here in stems, the bundles are always conjoint, means xylem and phloem are seen in a single vascular bundle, joined together. Then collateral. Collateral means xylem always towards the inner side and phloem towards the outer side of the bundle, and both in a same straight line. So it is called as collateral. And then open. The bundles are open. That means in between the xylem and phloem there is cambial tissue present. Because of the presence of cambium, the bundle is open to secondary growth. So the vascular bundles of a dicot stem are conjoint, collateral, and open. Each vascular bundle is wedge shaped and it consists of an outer phloem and inner xylem. In between there is a cambium. Xylem is endark, means the small protoxylem elements are always facing the central portion of the stem or the pith of the stem. So such a condition of xylem is called as endark xylem. Xylem is made up of tracheids, trachea or vessels, xylem fibers, and xylem parenchyma. Protoxylem elements are at the center and metaxylem towards the periphery. The phloem consists of sieve tubes, companion cells, phloem parenchyma, and bast fibers. Cambium is vascular meristem and occurs in between xylem and phloem. It is also known as intrafascicular cambium. Fascicle means bundle, Within the bundle, the cambium is present, so it is called as intrafascicular cambium. These cells are rectangular in shape and arranged in 3 to 4 layers. Then coming to the medulla. We finished with pericycle and the vascular bundles. Now coming to the medulla or pith. It is the most conspicuous region of dicot stem and consists of a mass of parenchyma cells with intercellular spaces. You all can see in the ground plan of the dicot stem, there is a wide pith in the center within the vascular bundles. So that is the most conspicuous region of dicot stem, and it consists of a mass of parenchymatous cells with intercellular spaces. Now lastly coming to the medullary rays or pith rays, which are found in between the bundles. In the interfascicular region or in between the vascular bundles, the parenchyma cells of the pith, central pith, extend radially and connect to the endodermis. These are known as medullary rays and they serve to store food materials. So the medullary rays are nothing but the parenchyma cells of the pith which run in between the two vascular bundles and go to join the endodermal cells. So this is all about the dicot stem. Now coming to the monocot stem. Here example is the maize stem. The TS or transverse section of a monocot stem can be studied with the help of a thin transverse section of maize at internodal region. The section appears more or less circular in outline and shows three fundamental tissue systems, namely epidermis, ground tissue and vascular tissue. The epidermis. It forms the outermost boundary and is composed of a single layer of compactly packed, narrow, rectangular or barrel-shaped cells without intercellular spaces. A few stomata may be present in young stems, but epidermal hairs or trichomes are completely absent. The outer wall is cutinized. Cutinized means there is deposition of cutin outer to the epidermal walls, epidermal cells. So this is about the epidermis. So here there are no trichomes or the multicellular stem hairs which are present in the dicot stems are absent in monocot stems. Then coming to the ground tissue, the next zone is ground tissue. In monocot stem, there is no differentiation of zones like cortex, endodermis, pericycle and pith. So all these four regions are absent in monocot stems. Instead, there is the entire massive region known as ground tissue. Below the epidermis, you can find a ground tissue which is differentiated again into outer hypodermis and inner parenchymatous zone. The hypodermis is just beneath the epidermis. The ground tissue here consists of 3 to 4 layers of thick-walled compact sclerenchyma cells. So, in the ground tissue that is just beneath the hypodermis, the ground tissue is made up of sclerenchyma cells, 3 to 4 layers of thick-walled compact sclerenchyma cells. This sclerenchyma may be interrupted by parenchyma at the region of stomata. Then comes the wide parenchymatous zone. Below the hypodermis, the ground tissue is composed of thin-walled parenchymatous cells with intercellular spaces. Then coming to the vascular bundles. In monocot stem, a separate stelar region is absent. There is no stele. As I told you earlier, there is no cortex, there is no endodermis, there is no pericycle and there is no pith. While in dicot stem, there is a cortex which is differentiated into 3 zones, and then there is a wavy endodermis, and then there is a parenchymatous and sclerenchymatous pericycle, and then there is a wide pith. But here all these 4 zones are absent. The vascular bundles are numerous in number and they are irregularly scattered in the ground tissue in monocot stems. The peripheral vascular bundles, or the bundles which are nearer to the epidermis, are smaller in size and arranged densely. They are more in number and smaller in size, while those towards the center are larger in size and they are less crowded. Large number of few vascular bundles are seen in the center of the monocot stem. The vascular bundles here are conjoined, collateral and closed with endark xylem. So here also the bundles are conjoined, means both xylem and phloem are seen in the same single vascular bundle. Collateral, xylem towards the inner side of the bundle, phloem towards the outer side of the bundle, and both in the same plane. And closed means there is no cambium present in between the xylem and phloem. That means the bundles are closed to secondary growth. There is no secondary growth occurring in monocot stems. And the xylem as usual is endark, means the protoxylem is facing the central part of the stem and the metaxylem faces the periphery or the epidermal region, outer portion of the stem. Each vascular bundle in monocot stem is oval in shape and looks like a human skull with the two eyes, nose and mouth. So it is, each vascular bundle is surrounded by a sclerotic bundle sheath, a bundle sheath made up of sclerenchymatous cells. The xylem here is arranged in the shape of the English alphabet capital Y. There are two large metaxylem vessels, that is one occupying each arm of the Y, alphabet Y, and two small protoxylem vessels which occupy the limb of the Y. And there is a narrow cavity, and the lowest protoxylem vessel usually disintegrates, dissolutes, forming a water containing cavity called as lysigenous cavity. It is also called as the protoxylem cavity. The two metaxylem elements look like the two eyes of the skull, eye sockets of the skull, and the protoxylem elements look like the nose, and the lysigenous cavity looks like the mouth portion of the skull. And then in between the two metaxylem elements, just above the two metaxylem elements towards the periphery, is found the phloem tissue. The phloem lies outside the xylem and consists of sieve tubes and companion cells only. Phloem parenchyma is absent in the phloem of monocot stems. The protophloem or the primary phloem or the first formed phloem forms a narrow broken band near the periphery of the vascular bundle. Cambium is absent in monocot stem. So this is about the anatomy of a monocot stem. So this is the explanation of the monocot stem which I already told you. So the bundle, you can have a closer look of the skull shaped bundle, monocot bundle. Xylem is Y shaped, two metaxylem arms of Y and two protoxylem limb of Y, and then a lysigenous cavity or a water containing cavity. And phloem in between the two metaxylem elements. Phloem is found. Now coming to the differences between the dicot and monocot stem. Dicot stem, there is hypodermis which is made up of collenchyma. In monocot stem, hypodermis is made up of sclerenchyma. In dicot stem, the ground tissue or cortex is differentiated into outer collenchymatous, middle chlorenchymatous and inner parenchymatous and innermost endodermis. In monocot stem, the ground tissue is undifferentiated, or it may be just differentiated into a hypodermal sclerenchymatous cells and general ground tissue made up of parenchymatous cells with intercellular spaces. In dicot stem, starch sheath or endodermis is present which is wavy. In monocot stem, starch sheath or endodermis is absent. In dicot stem, medullary rays or pith rays are present in between the vascular bundles. In monocot stem, there is nothing such as medullary rays, or the medullary rays are absent. In dicot stem, a pericycle is present which is partly made up of parenchyma cells in between the vascular bundles and partly made up of sclerenchyma cells above the vascular bundles. In monocot stem, pericycle is totally absent. In dicot stem, a wide pith is present. In monocot stem, there is no pith. In dicot stem, the vascular bundles are in the form of a broken ring and they are arranged in a proper manner, and the bundles are open. There is cambium in between xylem and phloem. In monocot stem, the bundles are scattered irregularly and the bundles are closed. There is no cambium present in between xylem and phloem. The proper arrangement of vascular bundles in dicot stem is called eustele, and the irregular scattering of the monocot vascular bundles is called as atactostele. In dicot stem, there is a bundle cap present. The sclerenchymatous pericycle forms bundle cap above each vascular bundle. In monocot stem, bundle sheath is present, which is sclerenchymatous. In dicot stem, protoxylem lacuna is absent. In monocot stem, protoxylem lacuna or water containing lysogenous cavity is present. In dicot stem, phloem parenchyma is present, and in monocot stem, phloem parenchyma is absent. This brings us to the end of this session about the dicot and monocot stems in detail. So coming to the conclusion, we studied about the epidermis, we studied about the cortex or hypodermis in both the stems, we studied about the endodermis or ground tissue, we studied about the vascular bundles which are conjoined in stems, collateral in stems and endarc in stems, and they are open in dicot stems and they are closed in monocot stems. And the pith is present only in the dicot stem but not in monocot stem. These are the references. Thank you.